क्या? प्लीज अटेंड। कब है ये? वही मैं बोला ना, मीटिंग है मेरे। Okay, good evening, everyone. I think we should get started now. So, welcome to week 8 of Bioinformatics Algorithms and Applications. So, today we'll be mostly talking about mutations and how these mutations uh, change protein structure and function. So, excuse me. Yeah, so what if changing a particular residue, amino acid residue in the whole protein, how changing one residue can uh, impact the function and the structure of the protein, and in some cases it does not. So what kind of factors are associated with mutations like these, and why should we be more concerned about this kind of mutations? Because a lot of these examples that we are going to talk about, say for example, sickle cell anemia, valine to glutamic acid to valine. It's a simple amino acid change, but it's a single amino acid change, but still causes like a very impactful disease, uh, which has a lot of prevalence and causes a lot of deaths per year. So small changes in certain cases can bring about these changes in the whole structure and function of the protein itself. In some cases, it changes. It does not allow the protein to fold correctly in its own manner. The protein folding becomes wrong, and hence the protein becomes not usable for the cell, and it is destroyed by pathways like ubiquitin and other things. Right. So, uh, yeah, these mutations are important, but again, if it is happening in a gene of very high importance, mostly what will happen is at least it'll be a recessive inheritance, as in like one copy the of the mutant protein won't actually cause the disease. You need to have two copies. Both the alleles need to be mutated in your genome to get to get the both the copies mutated and a um, like non-functional protein. Right. In some cases, haploinsufficiency occurs where basically one copy of the mutation is enough to cause the disruption in protein structure and function. And in some cases, uh, um, yeah, in some cases, these mutations can cause the disruption in in the structure and the function. In some cases, it does not, because if you replace if you're talking about substitution mutations, if you replace an amino acid with another amino acid with very similar properties, say for example you're replacing a hydrophobic amino acid of a certain size with another hydrophobic amino acid of a very similar size, then it becomes almost the mutation becomes silent and almost what we call as synonymous mutations, right? Where where the amino acid change type is the same, like so those are known as synonymous, and where you have different categories of an amino acid changed. So maybe a hydrophobic to a hydrophilic, a polar to a aromatic, something like that. If those kind of mutations happen, those are known as missense mutations, and those missense mutations are the major cause of disruption of protein structure and function because it is changing the type of residue at that position at that particular for that particular gene or for the particular protein, right? So uh, yeah, mostly these set of mutations are going to be missense, and the synonymous are going to be the ones which do not actually cause this change in residues. Now, these mutations uh, can be classified majorly into two groups. One is a gain of function, one is a loss of function. Again, like it is these are very loose definitions. Mostly, the definitions that we talk about are mostly loose definitions, and it is not uh, the directionality does not always tell you about the impact of the disease itself. Say, for example, a loss of function variation or loss of function mutation in a protein might be less harmful or less clinically significant than a gain of function of the same protein. So LDLR is a good example of that. I think PCSK9 also has certain examples of this sort. So basically, you have these gain of function mutations which causes disease, but loss of function does not. So again, as the name suggests, loss of function is basically it's a complete loss of protein. Either the protein is malformed or the protein is not formed at all. So the cell is not being able to utilize the protein for its development, right? So it is also known as null variant or loss of function variant. And in case of gain of function, is basically you increase the expression of the gene or increase the production of the protein due to that variation, right? So uh, and it also can be acquisition of a new function or a ectopic expression of a function. We have this hypermorph or neuromorphs in basically a lot of cases in Drosophila, where you have these Antinapedia mutations, you have these. other set of mutations where the gain of function occurs, where basically, so not going into much details, but you have Drosophila is made up of a lot of segments, right? So every segment has some polarity genes which give the segment its features. And in each segment, either the segment will form wings, or it will form antenna, or it will form legs, or whatever, depending on the position of the segment, right? And the gene expression is controlling all of this. Now, what can happen is, in some cases, if some genes are having gain of function in certain segments of the fruit fly, what can happen is, like, you'll see that function being replicated in that particular segment, although that segment is not responsible for that function. So, for example, in if you um, if you express certain genes, if you express a gain of function mutation of certain genes in the like like, I think the second or the third segment. I'm not completely sure about the segment number, but near the head, if you express that gain of function mutation, what will happen is, you instead of antenna, uh, legs come out of from that segment. So from the head segment, antennas are supposed to come out, but in that gain of function mutation, legs come out, because you have conferred that property by introducing that gain of function mutation in that segment. So there's a lot of such examples of gain of function mutations in terms of both uh, disease-causing gain-of-function mutations, and also uh, stuff like these, where you can experiment with fruit flies in terms of, like, what if you do not know the function of a particular protein, either loss of function mutation or gain-of-function mutation can actually tell you about the function of the protein. So yeah, just wanted to make it clear that it is not that loss of function is bad, gain of function is good, or vice versa. It can be either, depending on the context. So always keep that in mind. So again, this is an example for gain of function. It is the beta globin gene, which mutation of which causes sickle cell anemia, right? It is inherited as a recessive trait, because if you have one copy of that variant, you are not going to be expressing that variant. But if you have both copies together, it is possible to be expressed. Uh, 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 yeah, but in case of inherited resistance to malaria, it is a dominant mutation. So it's the same mutation, but it is recessive when it is when the disease we are concerned about is sickle cell anemia, but it is dominant when we are talking about diseases like features like inherited resistance to malaria, right? Again, like, it is a very kind of a two-way street. Is basically, if you have sickle cell anemia, your hemoglobin cells become sickle shaped. Uh, they like, lose surface area to carry oxygen bound to hemoglobin, and because of the sickle shape, and so basically less amount of oxygen is carried by hemoglobin, so uh, it the person becomes anemic. But again, if the surface area is lesser for the hemoglobin molecules, what will happen is the plasmodium virus that causes malaria is its life cycle goes through the human RBC. So it has a full life cycle. It has a full cycle of life inside the human RBC. But if you have sickle-shaped RBC, it is not able to fulfill its cycle. So it gives you resistance to malaria. On the other hand, it gives you anemia as well. So these are kind of depending on the context. It it is either a harmful variation or a good variation. Again, depending on the context. Right? Okay. Now we're going to.
So diseases like hemophilia is loss of function, right?
Sorry.
Diseases like hemophilia is loss of function, right?
It is loss of function, but it is, I think, mm, just let me check quickly. I do not remember off the top of my head. Mm, it's X-linked disease. Yes. Mm, it's an inversion. It's not exactly uh, loss of function or a gain of function. So all mutations cannot be categorized into loss of function or gain of function, right? In some cases, it just affects the promoter of the protein, or in some cases it affects regulatory regions of the protein. It can affect a lot of things where the protein itself, you can see the effect of the mutation on the protein itself, but it's not always a loss of function or a gain of function directly. So in this case, it's something like that. So this mutation is not it's not a single substitution or a single deletion. This is an inversion. So a chunk of the chromosome has been inverted. It's a structural variant. It's not a single mutation. Right.
Okay, sir.
Yeah, yeah. Are you saying something?
No, sir.
Oh,
I was interested because I have to do a project report next week for my college on this
okay,
gene therapy, you know. So I was
okay.
interested about the disease.
Yeah, this does not look like a loss of function mutation. Uh, a good loss of function, I think there's a uh, so I don't know if uh, so I'll just quickly show you. So for example, this is the Nomad database. If you go for RS334 is the sickle cell anemia variant. I think T2G, no, T2A is the sickle cell anemia variant. Yeah, so this is not a loss of function, direct loss of function variant. This is a missense variant in the HBB gene, which causes that change, right? So not all uh, uh, disease-causing mutations are loss of function or gain of function. This is neither. This is a missense variant. Missense is just changing the amino acid at that position, and it is causing beta thalassemia, malaria, and all those things. Like right, but this is not a loss of function or gain of function uh, mutation, right? And it is, if you see the distribution in different ancestry groups, it is more common in African American individuals compared to the other uh, ethnicities as well. So that is also the mutation effect of the mutation is also specific to certain groups of individuals who carry that mutation and other mutations that are linked. So it is so in cases like sickle cell anemia and say other monogenic disorders, the disorder is caused by a single mutation, which if you know the mutation and you know that this causes the disease. But in many cases, it is polygenic. So there's like not one single mutation is causing it. It's like a host of mutations combined. The combined effect of those mutations is causing the disease. So what can happen is, along with this particular variant, there can be other variants that are inherited together because they are in the same recombination block in the genome, and so you can have more of these disease mutations coming inherited from the parents. Now what can happen is, just this particular variation might not be a loss of function variation, but other variations that are there in the genome that are surrounding it, combined effect of all of them might be a loss of function, right? So all I'm telling you all of this just to say is there's a lot of like a lot of kind of what to say variability in terms of how we see mutations and what kind of effects those mutations are causing, and like these mutations are also not equally distributed along the human race. Some ethnicities have more, some have less, depending on different practices and different inheritance patterns, right? So yeah. Okay, coming back, we're going to talk about some specific uh, mutations. So one is the alanine to valine. So here what you're doing is basically alanine is a much smaller residue than valine. So you're replacing uh, a smaller amino acid residue with a larger one. So what can happen if it is in a turning point in the fold of the protein, or in a very dense region of the fold of the protein, this substitution can cause irregularities in protein folding, because you're replacing an amino acid which takes up smaller space with an amino acid which requires a larger space, right? So it is, if the local environment has available space, this can create a well-packed structure, and then that can lead to gain of additional contacts, additional folding, additional interactions with other amino acids. But it is, if it is already in a dense region, in a tightly packed region, introducing a larger residue will cause steric hindrance, and it will destabilize the protein, right? Similarly, and the concept is vice versa for the opposite thing, which is valine to alanine, where you are taking out a bigger residue and putting a smaller residue there. Uh, what can happen is this can cause cavities inside the protein 3D structure, and it can it can allow certain movements within the fold, and this loss of tight packing interactions will generally disturb the stability of the fold. So if you are directly substituting a particular amino acid with another amino acid which is not similar uh, to the one that you're replacing with, in terms of chemical properties or in terms of physical properties, if either of them are not matching, it can cause disruptions in protein folding. It can cause disruption if say, for example, these set of mutations that you're doing it on the active site of an enzyme. So that means the enzyme might not work after that. So uh, depending on where the the mutation is happening and the context of the mutation, like, is it happening in a denser place, it is happening in a less tightly packed space, is it happening, are you changing the residue on the surface with some, so if on the surface of the protein you are expected to have hydrophilic and polar residues, right? If you're replacing one of those hydrophilic or polar residues with a hydrophobic residue, it is not going to sit well there. The protein folding will not happen that spontaneously, because the chemical properties of the uh, amino acids on residues on the outside of the protein has changed. So this slide is there to tell you what kind of physical and chemical changes a particular mutations can bring, right? We look into other type of mutations, for example, alanine to aspartic acid. So this is basically, there's a change in the electrostatic charges that is going to be present in this amino acid residue versus the one that you are going to bring in. So it replaces an uncharged amino acid with a charged one, which can significantly alter the local electrostatic landscape. And introducing a large charged residue into a buried hydrophobic location is likely to severely destabilize the protein due to unneutralized charge. As I was saying, that if you are if the mutation is happening in a place where it is where you are changing an uncharged or mutating an uncharged amino acid to a charged or polar amino acid. Uh, uh, but if the location of that amino acid is within a dense hydrophobic residue, set of residues, it can definitely cause disruption in the structure, because uh, again, like, these hydrophobic residues will be present inside, present within the folds. So if you are introducing some amino acid, charged amino acid there, because of the charge present, the folding will not happen properly, or uh, the amino acid won't be buried inside because it is charged, so it by nature would not behave like a hydrophobic residue, right? So in terms of space, in terms of charge, uh, both disruptions can happen if you substitute a particular type of an amino acid with another type. And also it can also change the structural integrity, because again, in a hydrophobic location where it is very important for the protein to fold, and all those things, if you introduce a charged amino acid there, it is not going to fold or function properly. Other type of issues that can happen is basically if you do the opposite. So basically, if you're removing a charged amino acid and replacing it with an uncharged amino acid. or an aliphatic one, which is in this case leucine. So it what it can do is basically, because of the charge of the amino acid that was initially present, it would have made hydrogen bonds, it could have made ionic bonds, bridges, and it was interacting with a lot of other residues, because mostly because it is charged, right? So what can happen is that these extra bonds, formation in terms of ionic bonds and hydrogen bonds, will not happen if you keep an uncharged and a non-polar amino acid there. So again, it is going to have some structural impact, because these ion pairs often act as critical structural anchors. Severing them disturbs the structure and destabilizes the protein. So these two slides are kept here to kind of give you an idea what basic disruptions can happen when you when mutation occurs, right? What are the physical and the chemical possibilities? Right? Now, there comes the concept of stabilizing residues. So, what are these? So, these are specific amino acids uh, within a protein that disproportionately contribute to maintaining its 3D fold. Uh, and while every amino acid plays a role in the protein structure, stabilizing residues are the load-bearing pillars, keeping the protein thermodynamically stable and prevent it from unfolding. So basically, these are the again, like as the line says, that if all the amino acid residues actually contribute some way or the other to the structure of the protein, right? But there are some amino acids which will have a bigger role in conforming to the structure than some other amino acids. Some amino acids will have more specific functions in terms of binding to other targets or other proteins that can happen, but some amino acids are there just to act as pillars for the whole folded 3D protein state. So you have a few different category of amino acid residues here. So first is the hydrophobic core. So we have discussed hydrophobicity in great detail in earlier classes. So if inside the protein uh, where these folds are occurring, where you have amino acid residues buried, mostly it will be your hydrophobic non-polar residues, including leucine, isoleucine, valine, etc. Right? So these amino acids cluster together inside the protein interior, and these residues minimize solvent accessible surface area for non-polar groups and significantly lowering the free energy of the folded state. So these hydrophobic residues are quite important in terms of maintaining the protein structure, because these make up the hydrophobic core which gives the protein its basic structure. Then you have salt bridges, where the, basically these are ionic bonds that form between polar residues. These are strong electrostatic interactions between oppositely charged amino acid chains. So you, in the positively charged categories, you have arginine, lysine, and histidine, and for negatively charged, you have glutamic acid and aspartic acid. Uh, while often found on protein surface interacting with water, salt bridges that are buried deep within the protein interior or located at the surface interfaces of multi-subunit protein complex provide massive stabilization. So these are very important. This salt bridges, formation of the salt bridges because of the presence of this charged amino acid, gives the structure the stability that it requires. Right? Now, there can be, so these are the major ones. There can be other factors involved as well. So first is the hydrogen bond formation. Hydrogen bond, although is very a weak bond, but still it stabilizes, gives stability to the protein. So you have polar residues contributing to this, because bond formation is between the where the double-stranded oxygen, sorry, not double-stranded, double-bonded oxygen actually pulls one electron from the hydrogen, forming a temporary bond. This is the hydrogen bond, right? Uh, so you need a polar residues to create this hydrogen bond. So residues like serine, threonine, asparagine, glutamine, tyrosine, etc. Uh, uh, these are residues that form. hydrogen bonds. So either you need to have the free hydrogen or you need to have the bonded oxygen free for bond formation. Right? Mm, The stabilizing residues often form extensively cooperative hydrogen bond networks, although these are very weak. But if you form a network of these bonds, these together become very useful. A single residue might act as both the hydrogen bond donor and an acceptor, locking different secondary structural elements like alpha helices and beta sheets together. Then you have cross-linkers. Cross-linkers are mostly we are talking about sulfur bonds. So basically, it's a covalent disulfide bond, and it is a very strong bond. Sulfur-sulfur bond is, I think, smaller than the oxygen-oxygen bond, so it is stronger uh, uh, than two than oxygen bonds, and so these act as kind of backbones inside the protein. These are very strong bonds which confer the shape of the protein. And there are two amino acids mostly involved. The main one is cysteine, but also methionine also contains sulfur, but like it cannot directly form covalent disulfide bonds like cysteine. So cysteine is the major player here, where it can form these cross-linking bonds. They contribute much more to stability uh, in terms of like, so hydrogen bonds, you need a lot of bonds to form to give you that stability, but sulfur bonds are so disulfide bonds are so strong that much lower number of the same bonds can give you better stability. Then finally you have conformational restrictors, like, so these are amino acids that stabilize the protein not by interacting with other side chains but by dictating the shape of the protein backbone. Right? So proline amino acids, sorry, amino acids like chlorine and glycine, they have these ring-like structures which restricts backbone flexibility, reducing the entropy of the unfolded state and locking specific structures in place like turns. Right. Conversely, glycine lacks a side chain, allowing it to fit in incredibly tight spaces. Glycine is the smallest amino acid, right? Allowing it to fit in incredibly tight spaces where any other residue would cause steady clashes and destabilize the protein. So these are basically unique characteristics of amino acids that give certain unique features to the proteins. So changing this, so glycine is basically the USP of it is it is very small. It can fit anywhere, and if you mutate it, generally the mutated amino acid will be larger than glycine itself, and it is not going to sit well within the whole structure because glycine itself takes very small amount of space because it's the smallest amino acid. Okay. Uh, okay. I'll come to discuss this. Just give me 1 second. I'll do that. I hope I'm audible. Okay, yeah. So we are discussing about different types of amino acid residues and what they confer. Like if you mutate particular types of amino acid, what issues you might face, right? What issues the protein might face in terms of folding. So it can be like you're replacing chemically less. Sorry, I'm audible, right? And you can see my screen. Yeah, thanks. Yeah. So for example, if you are changing the chemical property of the amino acid itself, you are replacing a charged or a polar amino acid with a non-polar amino acid, or vice versa. Or if you're changing the physical characteristic of the amino acid that is present, if you are replacing a smaller amino acid with a bigger one, or a bigger with a smaller one, or you are replacing amino acid which do particular functions, like for example, cysteine will form those disulfide bonds. If you're replacing cysteine with anything else, it will not form the disulfide bonds, and it will not create a good structure of the protein. So again, all in all, like if it is not a like for like substitution, if you're substituting, if a mutation is a substitution mutation, and if it is not a like for like, a similar type of amino acid being replaced by another similar type of amino acid, then all the variations are going to produce either misfolding or some mutant form of the protein, or like in some cases maybe no protein at all, right? So uh, yeah, so if you have synonymous mutations, so there comes the concept of evolution. Here is basically if you are thinking about synonymous mutations, where you are replacing a similar type of amino acid with another similar type of amino acid, you are not changing any structure or any function of the protein per se. In some cases it might change, but in most of the cases it won't change anything. So that is in those cases, uh, so synonymous mutations you'll see a lot more compared to missense mutations in the human. I'm talking about human genome, but in general in any genome, because synonymous mutations are much more tolerated from an evolutionary perspective, that you're not changing the amino acid recipe, you're not doing anything per se functionally or structurally to the protein. So in most cases, those synonymous mutations are going to be uh, propagated to the next generation. But a lot of missense mutations, what can happen is can cause diseases like sickle cell anemia. So what you see is a less prevalence of these missense mutations compared to synonymous mutations. So it is almost like for 2 to 3 synonymous mutations, you're going to see one missense mutation, which is quite fascinating and quite intuitive as well. Because yeah, from an evolutionary perspective, the selection is much harder on the mutations, missense mutations compared to the synonymous ones. Right. Now, if you ask what are the, like, in general, what forces act within a protein and between proteins and solvent molecules that would cause a protein to fold spontaneously to a unique 3D structure. Like, what are the forces involved? So it can be a host of things. It can be long range interactions like ion-ion, ion dipole, 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 like charged interactions. It can be short range, van der Waals forces and other attractive forces. It can be local between adjacent amino acid in a linear sequence, or non-local between sequences separated in a linear sequence, meaning like it is far away if you think of it in a linear sequence, but in a 3D structure, they come close together because of some folding or something else. Uh, Clues as to what stabilizes the tertiary structure of a native protein can be gained by subjecting the proteins to agents that unfold or denature proteins. So if you just uh, uh, expose a particular protein to a denaturing agent, and you know the property of the denaturing agent, what that denaturing agent is actually doing, so then you can understand uh, what are the forces that are actually stabilizing the structure, and this particular uh, denaturing agent, what is it hampering with so that the structure is not functional after treatment with the denaturing agent. So uh, experiments like these have been conducted previously and still continue to do so. So some well-known causes of denaturation we know as pH, extreme pH, at like at a I don't know if you remember or not that particular curve of temperature and pH. So at a particular temperature, at a particular pH, the protein is the most stable. And if you move away from that temperature and the pH, it becomes unstable. So pH is a big deal in terms of determining if the protein is stable or not. Like it is stable at a certain pH, but it will not be stable at other pHs. High concentration of some salt solutions or organic solvents and temperatures. So temperature and pH are the most basic ones, and then you have chemical reagents which can denature proteins. Uh, so if you do such experiments and you do thermodynamic experiments like that, and what you will find is that native proteins are only marginally stable, about 4.4 kilojoule per mole per amino acid, or about negative 10 kilocalorie per mole. So this for a protein of almost 10,000 molecular weight, 10,000 Dalton of molecular weight. And this is quite weak, right? So the stability energy that you have, sorry, the folding energy that you have, which is right around 42, or sorry, 10 kilo, negative 10 kilocalorie per mole, it is equivalent to almost two hydrogen bonds. So it is not very strong because there are other factors that are involved uh, uh, that is causing the uh, increase in energy. So all this and all these interactions, hydrophobic interactions, hydrogen bond interactions, van der Waals interactions, they bring down the delta G of the protein because they are more stabilizing the protein. They're interacting with each other. They're folding and they're stabilizing the protein altogether. But there is something called chain conformational entropy, where which increases uh, uh, the delta G of the protein itself. So the um, net delta G, basically the net energy that is stored because of the stable protein, is basically it's quite low. It's almost at the level of two hydrogen bonds. So it is very easy to destabilize a protein. It is very easy to denature a protein. It is not that difficult. If you proteins exist in these 3D states under certain specific conditions. If you change the pH slightly, if you change the temperature slightly, if you expose it to some denaturing agent, it will denature it very readily. It is not uh, it is not that stable per se. And this chain conformational entropy is basically a thermodynamic quantity. That is how it is defined. And it is associated with the number of possible spatial uh, conformations of that protein. Right. So uh, uh, it is basically, yeah, it is an entropy that directly affects the stability and the flexibility of the protein. And it is high in disordered or unfolded state, and it decreases when chain folds or are restricted. Protein folding is happening, and typically, uh, like it is almost equal to the delta G that is produced by the hydrophobic effect, the hydrogen bonding effect, and the van der Waals effect. So the purpose of this slide is to tell you that denaturing a protein is not that difficult because the delta G stored in that are stored because the protein is folded. Basically, if you subtract the Gibbs free energy of a folded state minus an unfolded state, it's not that different. So it's only at the level of two hydrogen bonds of a protein of molecular weight 10,000. Right. So how do you compute the stability of the protein? How do you say that this folded structure is more stable than that structure and vice versa, right? So we use certain metrics. So all of these metrics mostly we have discussed earlier, but I'm going to go through them a little bit. in a little bit. So surrounding hydrophobicity. So as you remember, there's the hydrophobicity scale. So every amino acid will have a certain hydrophobicity associated with it. So if you take like around 20 residues in and around the residue that you are working with, so it will be the average hydrophobicity of all the residues, of all the 20 residues that you are working with. Right? Then you have long range contacts, stabilizing centers, and other conservation scores that we look at, right? So surrounding hydrophobicity, as I was saying, it's basically the average hydrophobicity of all the amino acid residues in the surrounding of the residue that you are interested in. So it is calculated as a weighted sum, where n, so weighted sum of nh, when n is basically the number of residues of type I around the central residue J within an 8 angstrom radius measured from the carbon alpha atom, and it and the hydrophobicity of these values are dependent on some scale. In, we talked about Doolittle scales earlier, but they can be other scales as well. So uh, yeah, so whatever experimental values or computed values from different models you can use to calculate the surrounding hydrophobicity. Right? Now, long range order, we know what it is. It is basically, although in the sequence, the two amino acids are very far away from each other, but due to the folding and due to the 3D structure of the protein, the two amino acids come very close to each other physically. So, uh, yeah, so it is given by the formula where you have these. This is basically small nij is basically the long range contact, and capital N is the total number of residues, and basically nij will be dependent on I and j, where these two residues in which C alpha distance between them is 8 angstrom. So basically, we take these two residues, uh, both the residues from C alpha should not be greater than 8 angstrom. So that is how they define it. Uh, so, and if a long range contact is defined to be, it's taken as a binary parameter. So either it can be 0 or it can be 1. So if the long range contact, if there is contact and they are within 12 base pairs of each other, then it is taken as 1, and if it is in contact with greater than 12 base pairs, it is taken as 0. So that is how they define it as a binary variable. Right? Other important factors are basically conservation scores. So uh, recall from sequence alignment and multiple sequence alignment, we talked about PAM matrices, we talked about closure matrices. So those determine basically how conserved a particular amino acid residue is at a position, right? Just 1 second. Just give me 1 minute. प्रियंका तो कल मीटिंग नहीं होगा क्या? नहीं, मैं पढ़ा रहा हूँ अभी, क्योंकि परसों छुट्टी है, उगाड़ी छुट्टी है, प्रॉपर प्रॉपर छुट्टी है. तो फिर मैं चला जाऊंगा सुबह टॉक अटेंड करने. हाँ, मैं खा. तो फिर आई एम अज्यूमिंग देयर इज नो मीटिंग टुमारो, एटलीस्ट 10 एम. और रजिस्टर करना पड़ेगा क्या इसके लिए? इसके लिए रजिस्टर करना पड़ेगा टॉक के लिए? ओ, हाँ, नॉर्मल रजिस्ट्रेशन, हाँ ठीक है, थैंक्स. देखते sorry for that, guys. Yeah. So there are other conservation scores also associated with it. Like we can look at. So B factors we have already discussed earlier. It tells you about the flexibility of the residue in terms of B factor. So if you have a less flexible protein, it is likely to be more stable. If you have a more flexible protein, it is likely to be more less stable. Sorry. So B normalized B factor is defined as like, so this is basically the Z-score normalization. So original B factor minus mean by sigma. So this is the Z-score normalization. And if it is greater than 1, it is flexible. If it is less than 1, it is rigid. So yeah, right. Now, there, these are computational ways. These are like mathematical or theoretical ways to gather information about the stability and basically flexibility of the protein. And but again, these are all based on some assumptions, right? But experimentally, it is it is much more robust to find out if a particular protein is stable or not stable. So experimentally, how would you find out? Is basically if you do some thermodynamic experiments and you calculate the delta G before and after folding, or after and before folding, whatever direction you want to go through. So experimental procedures are used to measure the free energy change of a protein upon mutation of amino acid residues. So there are a lot of methods by which you can mutate a particular amino acid residue, and then you can calculate the energy of those states. So the idea would be that the energy is calculated when it reacts with something, right? The protein reacts with something. So either you can dissolve it in some solvent, or you can make it react with bind it with some other protein, and so on and so forth. So there is a lot of ways to do that. So those molecular biology methods we are not going to touch here. But yeah, so there are a lot of ways you can do that. So these people, these particular studies and these particular enzymes, historical data collections were done on these set of uh, enzymes in these studies, which turned out to be very fruitful and very what to say, no, like helped us understand a lot. So one such database where you can find this change in Gibbs free energy values is this database called ProTherm. I think this has been discussed very extensively by Sir. I think there's one demo as well. I'm sorry, I can't give a demo because I particularly have not like extensively used this database. So it is better that you follow the demo showed in the course itself than what if I show something that might be wrong. So I will not try to do that. So yeah, please follow that. But yeah, it is a database that collects this protein stability experimental data. And it basically tells you what delta G is there, what is the delta G if you move from one state to another for that particular protein. Uh, and it links with other known protein databases like PIR, Sysprot, PDB, and so on and so forth. And the main thing it stores is basically thermodynamic data. So it includes data of denaturation by urea or guanidine hydrochloride. So these are known denaturing agents. So you denature the protein, and then you calculate the delta G. So right, at every point, remember one thing: the delta G can only be calculated where there is some reaction, there's some change. The delta is basically uh, change in energy that you're saying, right? It's not like you cannot calculate absolute energy, you can calculate only change in energy. So to calculate the change in energy, you need to make the protein undergo some reaction or some sort of interaction. Then you can calculate the delta G during that process, right? So yeah, in this case, you have denaturation delta Gs from urea and guanidine hydrochloride, and then you have thermal denaturation as well. So uh, at higher temperatures when denaturation occurs, what is the delta G, and so on and so forth. So it will track temperature, pH, buffer name, concentration, ion name, and concentration. So all the chemical information you'll get what is the in Gibbs free energy change if you move from if you undergo the protein in this particular reaction, and so on and so forth. So this database is quite interesting in terms of that. Uh, these are like experimentally calculated delta G values, which tells a lot about the protein in terms of like how stable is the protein inside the cell. Because like why would you need to know this about the stability of the protein, right? Because inside the cell also there are a lot of reactions going on. There is a lot of uh, changes in pH, changes in hormonal secretions, change in a lot of things, that is a host of things that is going on altogether inside the cell. The idea is to find that like if this particular protein is like this, is it because it has to withstand these set of chemical reactions, or these set of denaturing agents, or these set of temperature and pH imbalances. So through evolution, is the protein more capable of handling such conditions? So those kind of things are very important to study. And in terms of uh, protein stability is basically another very important aspect of it is basically drug designing. So the idea is that if you design any kind of drug, the drug has to go and bind to something. It can be a receptor, it can be a protein, it can be an enzyme, it can be a lot of things. But it has to go and bind to something. So and in most cases, the something is a protein. Like it can be a receptor protein, enzyme is also a protein, some other protein's transcription factor or whatever that is also a protein, right? So mostly it is going to be proteins. So knowing the stability of the protein that your drug is targeting is very important, because it should be able to target that protein and completely maybe uh, make it a loss of function protein, and not loss of function. It will not cause a mutation, but say for example, it goes and binds to the enzyme in a competitive inhibition manner, right? So your drug will come and bind to the enzyme so that the main substrates can't go and bind to the enzyme, right? So stability of the enzyme will be very important, and stability before and after binding with your particular drug is also very important. And if it is not stable enough, then like even if it's a promising drug, it won't work because the protein inside is not stable enough. So it is not being able to withstand the binding with the drug. It is disintegrating or doing whatever, and it is not helping you treat the disease. So this, why I'm telling you all this is because I want to emphasize on the fact that stability, the structure, and the conformation of the protein are the most kind of important proteins, important uh, parameters, properties of the proteins when looking from macro perspective. The sequence is very important. The sequence is what gives the protein its whole structure and everything. But at a macro level, where interactions is occurring, where different reactions is happening and everything is happening, the stability of the protein and the conformation of the protein, both physically and chemically, is very important. Right? So these kind of databases are very handy, very useful for different kinds of things. Okay, now we'll talk a little bit about the mutations that are happening. So, from a statistical perspective, there are 20 amino acids, and it can be mutated. So, one amino acid can be mutated to 19 different amino acids, right? So, 20 times 19, there can be total 380 possible mutations, right? So, at every position, there can be 380 possible mutations, and you can classify these mutations into stabilizing or destabilizing based on the frequency of these mutations, right? So, uh, yeah, so some amino acids will be more frequently mutated, some will be less frequently mutated, and what it will tell you is because the more frequently mutated amino acids probably are not that important to the structure of the protein, or like the mutation that is happening is like a like for like mutation. So a polar amino acid getting replaced by a polar amino acid, or a hydrophobic getting replaced by another hydrophobic. So it is telling you it's kind of, if the frequency is very high, either it's a like for like mutation where the protein structure and conformation is not changing, or it is happening on a residue that is not very important for the structure of the protein or the function of it. Right? Okay. So experimental stability data is also used to find the relationship between amino acid properties and protein stability upon mutations. And basically, delta P is calculated uh, as a difference between mutated and wild type. So depending on the values that you are calculating, you subtract it from what, right? Uh, these type of mutations, the hydrophobic and the varied mutations we discussed, are very important because they're again, if you talk about stability, these are the ones that give the most kind of stability. They are the most strongly correlated with stability. There's a positive correlation between the hydrophobicity and stability of the mutation occurring in the buried B strands. So if in the buried beta strands, in the buried residual sections, if a mutation occurs, uh, which is hydrophobic in nature, then it is this is highly correlated uh, with stability. So more the hydrophobicity in these regions where the uh, folding and like all the residues are buried, where if more mutations, more hydrophobic mutations occur there, it is most likely to be making it more stable. Uh, Uh, so there was this one study. So there's this inverse hydrophobic effect as well. So in cases of mutations occurring in exposed location in the protein, a negative correlation is seen, which is expected because hydrophobic residues on surface of proteins won't help the protein much, right? Because it has to be it has to have some sort of hydrophilic properties to interact with the polar environments. This particular study by Pacula and Straw reported that increasing hydrophobicity decreases the stability if the mutation is located in coil regions and exposed in a protein. So it, the study says that if the hydrophobic mutation, after mutating, the hydrophobic residue has occurred in coil regions or in exposed areas of the protein where you would expect more hydrophilic residues, then it decreases the stability of the protein, which is again quite intuitive. And again, the vice versa is also true. Right? Uh, on top of all this, on top of all this information, and including delta G, including delta G and other information in terms of stability, hydrophobicity, long range contact, whatever, uh, all this information, uh, we can come back to the most basic information, which is the sequence, right? Because the sequence is what is making the structure. Uh, though a single property correlation can be used to explain stability change, these features lack the information about the location of the mutation. So if you have a neighboring sequence, you have a wild type sequence and you have a mutated sequence. So you can take. Uh, I I think you remember how we calculated differences. We did not look for single amino acid. We look for a region of 20 amino acids. It's a region of it's a sliding window. It's a larger region, not a single amino acid. So that difference can also be quantified in terms of what is the wild type sequence versus what is the mutated sequence. In structure, in the case of structure as well, it can be quantified in terms of a lot of things. Uh, Say for example, average x coordinate positions, average y coordinate positions, average z coordinate positions of all the amino acids in the mutated state minus in the wild type state. So difference in structure and function is also important and needs to be incorporated. And this inclusion improved the correlation in partially buried and exposed mutations. So whatever insight that you're looking at in terms of, say, be it in terms of thermodynamics, the delta G, in terms of, say, specific amino acid mutations, in terms of stability between mutated amino acids, or in terms of like where the amino acid mutation is occurring in a hydrophobic residue or in a hydrophilic region in the surface or in the buried regions and whatever. The idea is that the sequence and the structure uh, is the driving force. The sequence is probably the most important driving force. The structure is also very important. So, given the structure, if you make a mutation, uh, then you can predict a particular thing. But if you do not know the structure and you think about the mutation, it is hardly going to predict anything, right? So, yeah, coming to the basic facts. Okay, so how do you predict the difference in stability? How do you predict the difference in conformation? The relationship between a property, it can be anything, it can be hydrophobicity, it can be structure, it can be sequence, whatever property that you're looking at. The relationship between a property delta P with stability parameter some delta G can be represented mathematically as linear equations. You can use multiple regression. So ultimately what you're trying to do is uh, like explain the change in use-free energy, right? How are you going to explain it? You're going to explain it through possibly long-range context. You're going to possibly explain it through some mutations that has happened, some other properties, right? So it is a multiple linear equation where your your dependent variable is your delta G, and all your independent variables are your independent parameters that you're looking at. Yeah. So, and if you take an energetic approach, because you know the delta G, you know the formula. It's RT, negative RT ln K, right? And this K can be represented by distance potential, right? So if you, it's basically um, distance between I and J before versus distance between I and J now, or it can be related to torsion as well. So directly not taking the distance, but taking some torsional force that is occurring before versus after. So all of these things can be represented as the delta G, right? Depending on what the change you think is happening, right? So all these things are taken into account. So your prediction model becomes something like this. So this is a linear regression model, right? So this is your uh, dependent variable, which is your delta G. This is the constant of regression. Then for all the amino acid residues, you're summing up the distance metric. I think this will be T. This is given wrong. And the torsional metric. And you're trying to regress it. And if you do a linear least square fit on this regression, uh, it yields a prediction accuracy of 85%. So using the distance potential and the torsional potential, you can predict the delta G with up to 85% accuracy. So that is also very, very fascinating kind of thing. Okay, I think, yeah. So I have a couple more slides left. Okay, so all these were related predictions of stability related to the structure, conformation, different bond formation, and other things, right? But sequence-based predictions again remain very important because I think we discussed a lot about sequence-based predictions in the last classes. Now, again, with the advent of tools like AlphaFold and everything, any kind of mutations that you can think of, you can predict what is the structure difference it is going to be, right? Uh, yeah, so you can, but earlier what used to happen is you can make a model, prediction model using features from these protein sequences. You can use the wild type residue, mutant residue, pH temperature, neighboring information, and you can make a user decision tree based method, and uh, it can gain an accuracy of up to 80% and correlation of up to zero point 0.7. So, uh, so if you do this kind of prediction, what will it tell you is basically if say for example you're changing an alanine uh, to a glycine, right? So if in the wild type as residue it is alanine, the neighboring residues contain glycine. Then if you mutate this alanine, what is going to happen is basically it is going to be a destabilizing mutation and can predict with accuracy up to 97%. Now, these kind of models were very useful back in the day because we did not have sophisticated deep learning and machine learning models. And now we have those things, and it is much more easier to predict using those things, and much more accurate, I think, in terms of predicting uh, the structure and the conformational changes that can be happening. Right. So yeah, these are basically some of the web servers that are available to predict stability and tell you if a particular mutation is stable or not. So it uses these set of thresholds in terms of surrounding hydrophobicity, long range contacts, conservation scores, and so on. Uh, and so this is one case study where they identified 957 stabilizing residues in 63 barrel proteins, sting barrel proteins, and they identified these residues to be the stabilizing residues, the residues which actually confer to the stability of the protein, not not like other residues which are having lower impact. So this particular paper has done this done an analysis on that, and say for example it has taken up a mutation which is the cis126 mutation, and it has mutated it and studied that it plays an important role for stability, and it was correctly identified by the computational method. So this particular condition actually has worked, and the stabilizing residues are also functionally validated in wet lab conditions. So that is also important. So it is a validation that your computation method has worked. So yeah, so I think
So this works only for Timbarel, right? These
so.
thresholds.
This specific to this set of proteins, obviously. For other types of proteins, you'll need to have different thresholds. It's dependent on the protein itself. So if you, since it's a specific type of structure, right? It's a barrel protein. You cannot use the same cutoffs or same thresholds for say a transmembrane protein, right? Or some sort of globular protein. So since the structure itself is completely different, and all these parameters, the hydrophobicity and the long range contact, everything will be different for different types of proteins, right? So yeah, this is specific for imbalance. Any other questions? Okay, and we'll move on to the assignment questions. Mm. Today's contents were a little bit shorter. I saw on the course structure as well. So this week I think the next week also the course content is a little bit shorter than the other weeks. So probably we'll finish a little bit earlier for these two set of weeks. Yeah. Okay, moving on. The questions are also very easy today. So I hope to you can answer quickly. Yeah. So this is basically the definition of long range order. So yeah, the idea is that you have these residues very close or very far away. Very far away in the sequence, but when the protein folds, physically they come closer to each other. So that is long range order. Okay, moving on. Again, the thing is that these exposed coils are on the surface of the protein, and these are on the outer side of the protein, right? So you need hydrophilic residues, you need polar residues in these regions, and mutating mutating the polar residues to become more hydrophobic, it is not going to help with the stabilization because it is not going to interact well with the polar environment and the solvents that it is exposed to. So, correct. Okay, why only two? This is a very easy question. Wait for another minute. Okay. Yeah, basically all of them are used. So we discussed hydrophobicity, we discussed long-range interactions and conservation. All of the three parameters are used uh, for identifying stabilizing residues because all three are important. So the question is, we are given free energies of these set of mutations, and we are asked which is the most tightly packed residue position. So the mutation that is generating the most difference in the highest delta G will uh, be basically the one that we are looking for. So this is changing alanine to a valine, so a smaller to a larger amino acid. Uh, if the original alanine was located in a very tightly packed region, the extra bulk of valine would cause severe steric clashes, resulting in a large loss of stability. And since this has the most negative value, so this is the most tightly packed position. So the important thing is the mutation itself, right? Alanine to smaller alanine to a very large value. So based on that, what is the delta G? And the highest delta G will be the, mm, yeah, the most packed position. This is quite easy. Proton. Yep. Moving on. Okay, let's see. One is three. Give one more minute. Okay, so the question is which type of residues are dominant in stabilizing residues? So the question is basically what is the most frequent type of amino acids in stabilizing residues? The answer is aliphatic, like where it is hydrophobic, where it is not uncharged. They tend to bury themselves inside the protein, inside the buried residues of the protein, and this packaging drives the hydrophobic effect, which is the dominant force in stabilizing the folded 3D structure. So hydrophobicity is the 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 thing that you're looking for. The more the bet the more hydrophobic residues you have in your core, the more stabilized the protein is. So we were seeing that hydrophobicity is a very good predictor of the stability of the protein. So the more the hydrophobicity, the more stable it is. So aliphatic amino acids, which are non-polar, non-aromatic, are basically hydrophobic, and they are the kind of most important. I think this was option B, and someone answered option A. Option A is not going to be there. Aromatic ones with ring-like structures and all, it's not uh, the most common type of residues in stabilizing residues. Okay, moving on. Okay. Yes. So I'll go back to this particular slide. Oh. So inclusion of the sequence and inclusion of the structure both improve the correlation, and R squared, basically the percentage of variance explained. So it becomes a better model. So both sequence and structure contribute to that. Okay. Yeah, wait for another minute. Okay, the question is that you are replacing glutamic acid in the vicinity of lysine. You are replacing the glutamic acid to value. So, glutamic acid is a charged amino acid, and lysine al also is a charged amino acid, but the charges are opposite. So, glutamate has a negative charge, lysine has a positive charge. When they are in the vicinity of each other, they will form electrostatic interactions or electrostatic bonds, which are known as salt bridges. So, mutating the charged glutamate to a neutral valine will remove this negative charge that the glutamate has. And valine is basically a hydrophobic amino acid, and it will destroy the ion pair. So, previously, the lysine glutamic acid or glutamate interaction is going to be removed. So, the ion pairs will be removed because you are removing an charged amino acid with a hydrophobic non-charged amino acid. So yeah, lysine is positively charged, glutamate is negatively charged. They were forming bonds. Now once you remove glutamate, there is no bond. Okay, moving on. So we have two A's, one B. Okay. Okay. The answer is A. It's going to be negative. It's basically final minus initial, mutant minus wild type. So 19.9 minus 22.2, not the other way around. I think someone answered a positive 2.3, but it will be negative 2.3 because final minus initial. Okay. Yeah, please keep this many like positive, negative things in mind because in exams you're going to get questions like this, and yeah, these are easy questions to answer. Yeah, this is very easy. I should wait for 1 minute. Yeah, basically you can use all of the above. Yeah, so that's it for today. Today's was a shorter one. Do you have any questions?
Sir, the question got solved, the one I said earlier.
Ah, yeah,
It
yeah.
it was yeah, it was my mistake that the digits were actually the same, but the unit was Armstrong square, so it was surface area. So
Oh, okay,
that's
okay,
why the lower the surface area, the buried the
okay.
amino acid was.
Yeah, yeah. The question, like it was slightly changed, right? Instead of
Yeah, it was just a difference of the unit of the
and
square. So it was,
okay.
you were talking about the distance and the sir was talking about the surface area.
Yeah, yeah, yeah. Then it's okay. Yeah. Yeah, these questions are not supposed to be exactly same like same what he discusses and what I discuss because I think we have access to different questions. So yeah, if any of the questions seem exactly same, they might not be exactly same. Some differences might be there. So yeah. Okay. Any more questions? Okay, then I'll close the session. Thank you for joining. We'll see you next week.
Thank you,
Thank you.
sir.
Thank you.